Hello everyone, this is Wahida Kazi. Hope you all are doing wonderful. Today I'm here to discuss a new topic. This is real-time PCR and cycle threshold value. What is RT-PCR? RT-PCR stands for reverse transcriptase polymerase chain reaction. This is a laboratory method to detect specific genetic material such as RNA, DNA of a pathogen present in the specimen being tested at the lab. It is one of the widely used and most reliable method to detect COVID-19 or SARS-CoV-2. It is called real-time RT-PCR because the lab tech can see the result on a real time. That means even when the process is ongoing, by using specific marker and most recently the fluorescent dye to see the result. In RT-PCR method, the first steps is to clean the sample. So as soon as the sample is at the lab will use some chemical method to clean the sample to get rid of extra fat, protein, or redundant material from the sample as well as from the virus and extract the RNA. This RNA is a mixture of RNA from the pathogen or the virus of interest as well as the RNA from the human substrate. The next steps is to produce complementary DNA from the RNA of the pathogen. Usually lab will look for target. The target is the genetic sequence or the base sequence of a RNA or the RNA fragment of the pathogen of interest. For example, for novel coronavirus, because it is one of the subtype of other human coronavirus, it must have a specific genetic sequence or the base sequence or the mutated base sequence that is important for us to know to identify or identify or or differentiate this virus from the other coronavirus or other kind of pathogen. The complementary DNA actually represent the original RNA that retrieved from the pathogen, and like the original RNA target sequence, the complementary DNA will have the same target sequence. And in these steps, this mixture of RNA extract or complementary DNA produced from the RNA extract and substrates required and other enzymes like heat stable enzymes like reverse transcriptase and a primer will be added to the RT-PCR machine. The primer is nothing but a segment of DNA that has the complementary base sequence as like the target. The primer has predetermined base sequences to identify the target of interest in the original RNA or complementary DNA sequence. If it match, then the target the primer will attach to the target, and primer also has fluorescent dye. So it will be easier for detect once it is attached to the target. So it will produce the fluorescent band in the auto radiology or the machine at the lab. all this in the RT-PCR machine, the machine will start run the cycle. There are several cycles, and sometimes it is predetermined by the lab, or also depends on the type of assay or type of test it will be run by the particular lab. And these cycles are usually two type in general. Is the heated cycle, or the heat cycle, and then cool cycle. The heat actually causes the disintegration or separation of the two strands of the complementary DNA. Thereby it will expose the target base sequence, so that this target base sequence can attach to the primer, which is the predetermined base sequence. The term CT value, cycle threshold value, came from the cycle of the machine, because to reach the threshold level of fluorescent or threshold level of target to determine, the machine need to run certain cycle. If there are enough target, then machine will require small number of cycle to detect the target that reach the threshold level. And if the sample doesn't have enough target RNA or the base sequence, then the machine required to run extra cycles, or the large number of cycles, to get the fluorescent that reach the threshold. So by knowing the cycle value of cycle threshold value of a test, it is easier to tell, like when if the sample has enough target that we are looking for. If the sample doesn't have enough target, that has different interpretation. One of them could be because the swabbing process, or the swabbing was collected in a time when the virus shedding is reduced, and it could be this kind of there are different interpretation for the cycle threshold value. I think I will continue in another lecture, and this is the basic discussion about RT-PCR and cycle threshold value. Hope you enjoyed the video, and thank you so much for watching. Have a great time.